So now what I'm going to tell you about is other very established, very well-known techniques for unsupervised mining that are a little different from the spectral methods we talked about earlier. So remember, the spectral methods were fundamentally based on the affinity matrix, but there was other ways of kind of clustering things and looking at kind of this affinity matrix. Another way is kind of this sort of mixture of Gaussians or looking at distributions, which is a little bit like that kind of k-means type of thing. So now we're going to look at some techniques related to that, in particular this what's called latent Dirichlet allocation. And so I'm going to tell you about this technique. It's a kind of distributional technique, a very different way of dealing with your data, but kind of comes to somewhat similar conclusions. And we're going to talk about advantages and disadvantages. So again, let me start by motivating the technique by sort of talking about where it kind of comes from, which is looking at words in documents, and then I'll go into the biological context. So what you might want to do is kind of think about a way of classifying documents. And you can think about, in kind of an app, if you're a statistician, you might think about each document kind of generating words according to the distribution of words in a document. So you have these different documents. And let's get the laser pointer out here. This document, has a lot about dogs and so forth, as cats and stuff, and Olympics. And you can kind of see that each of these documents is generating words according to the words that show up in the document. So that's a sort of simple way of doing it. But what is a kind of nice simplification is to think of the documents as being associated with different topics. So for instance, these first two documents are associated with animals. This one with the Olympics is associated with sports. And then each of these topics then has a certain proclivity to generate words. So the animals topic will actually generate the words like dog and cat. And the sports topic will generate words like Olympics and so forth, and corner or something. And now we can think about, when we think about documents, we can think about less about the words in them, but the topics that they represent. And this is quite a useful technique. And this is used, I should say, in analyzing biological text, but of course it's used analyzing text in general. And let me give you a little bit of the math of this type of thing. So here's how we kind of lay this out. We think of these alphas and betas are kind of hyperparameters, and we think about kind of having, for any given document, we have a distribution of potential topics in that document, okay? So if we have a document I, theta represents the distribution of topics. And we can pick from that distribution of topics in accord to how likely they are a particular topic, okay? And then when we pick a particular topic, if we're trying to generate a word, we can go to a corresponding vector that represents the word frequencies for that topic to actually pick a word. So we have a distribution over topics in a document. We have the distribution over words within a topic. We select our topic and we then put these two together to pick a word. And so we can kind of run through this what's called plate model thing to kind of pick topics according to these distributions, okay? And here's a little bit of a more of an urn-like sort of drawing here, where we imagine a person sort of picks, we have these topics, they have different chance of being picked, and they pick a topic according to these different chances, according to this distribution. More likely they pick the animals topic. Once they pick a topic, then that topic has a different distribution of words, and they're going to pick from this distribution of words, and then they'll get a word for the document, and they can repeat this over and over again. Okay. Now, one of the aspects of this is that the sort of hyperparameters of this, what's called Dirichlet distribution, I won't go into detail. They unlike say PCA, as I'll talk to you, you can end up with having a very sparse distribution of topics. If you select your hyperparameters in a certain way, you can end up with a very sparse description of data, and this is very useful for, sort of describing your data in an easy way, as opposed to having small projections onto lots of components. If you set your hyperparameters correctly, you can get it. You can I don't want to use the word project, but you can describe most of your documents or your data in terms of a limited number of topics. And that's actually a very useful thing. And that's one of the problems with PCA, is that you can often find the main components, but there's this long tail. Now, let me tell you, now that hopefully you understand how this works, let me go into a more biological example and try to motivate this biologically. Again, the key here is not, it's the math of the LDA, but it's also understanding how you could map this into a biological context. It's not so obvious. So let me tell you how you could do it. So here's a picture that shows the transcriptional regulatory network. So now we have each transcription factor, and the transcription factors regulate tons and tons of genes. I'm just showing transcription factors, that how they regulate each other. And I'm showing the factors in different conditions. So let's just say there's condition 1, cancer, condition 2, normal. And I'm going to show the edges that are present, say, in one condition, one color, and the edges in another condition, another color. And you can see there's a network change. You know, there's a kind of some edges that are only present, say, in the tumor condition, some edges that are only present in the normal condition. And there's a lot of rewiring of the network happening. Of course, you know, a lot of people find these networks very hard to look at. Initially, the hairball, now we have like the, I don't know, making a messy hairball, changing it all around it. Very hard to fathom what's going on. So let's think about how the topics could help us here and how the LDA could help us. So the basic problem we're looking at is we have all these transcription factors. They have all these genes they regulate, okay? And they're going to change their edges, okay? But maybe what's better to do is to not think about the transcription factors regulating a gene, but to think about the factors regulating a topic. Okay, so we introduce these latent variables, these intermediate variables here, okay? And then the topics in turn are associated with genes. So what are the topics? Now let's think coherently. Well, the topics might be something like a pathway. You know, so for instance, this thing could represent a pathway that has 2 different genes. This could represent a pathway that has these genes, right? And you know, pathway genes can be shared between different pathways, okay? That could easily happen. And now we think of the factors, this factor as mostly regulating this pathway and to some degree regulating this pathway, and not thinking of it regulating all these genes. And this provides a useful simplification because, of course, we can go from a tremendous number of genes, you know, say 50,000 genes, including RNA genes, or 20,000 protein coding genes, to 50 or 60 pathways, or some small number of pathways. So now you can kind of see where we're going to go in this type of thing. We're going to have a cell, okay? And in that cell, we're going to talk about, in a particular condition, we're going to talk about the transcription factors. And as opposed to talking about who they regulate, we're going to say, what's the distribution of topics for these in this particular cell, okay? Okay, so we have a kind of topic distribution, okay? And now we can, of course, you know, if we want to figure out the particular regulation for any gene, we can pick a topic, and then you know, so we can pick a factor and a topic, and then we can see which topics that factor would regulate, and then we could kind of build up, you know, a kind of we could build up from the distribution, kind of a typical cell with that thing. And so we'll have, if we can think about these different topics, which are different pathways, they're going to be associated with different genes, okay? And now what's key is if we have different cell states, okay, we can have different topic distributions, but now we can describe that complicated rewiring figure in terms of changes in the amounts of different topics. And so we can see this topic vector changing, and that kind of provides a compact simplification to think about the network change. Okay, now another technique. So this is another distributional technique. I'm not going to talk much about it because it's somewhat complicated. We've heard enough of these, but I just want to put it out there. There's yet more, there's a profusion of these clustering methods, but one method that is popular, I did want to mention, is called tsne, and there's another one even that follows on that called UMAP. It's often used for clustering single cell data, and one of the key aspects of this method is that it really only weights local distances and not global distances. So it tends to give very nice clustering where things are clustered together locally and they kind of get splayed over the page. And sometimes people just visually prefer that, particularly for single cell analysis. And so here's a typical single cell analysis picture where people are looking at different cell types, and these are different samples. And the figure makes these ones where the cell types get clustered together well. The distances between them don't mean very much, but they fit in nice clusters. And now I thought, I'm not going to tell you much about the mechanics of tsne. I just wanted to explain it there. I thought it might be good to just look at a few of these types of methods. So here, this is a single cell data set, say for melanoma. Seurat, I should have said, is a popular single cell analysis program that uses tsne, or actually it says tsne here. And so here's kind of what you get when you look at cell types here. And you can see that they kind of make these nice little bubbles that are separate from each other, okay? And on the other hand, if you look at PCA, if you just did SVD, it tries to keep the global relationships between things. And so these bubbles kind of merge with each other, and people find this sometimes less satisfying for dealing with single cell data. Now notice if I do, instead of using PCA, if I project the single cell samples onto kind of a set of reference components, and then I do RCA, notice I get some of that bubbly aspect to it. So I kind of, for some of the main groups, I kind of separate them well. So RCA is a kind of a, you might say, a halfway there to tsne. And a lot of these things for unsupervised clustering, you have to kind of try different techniques and see which one kind of gives you the best view of your data. Now, another comparison I wanted to show you was, I was talking to you about the sparsity thing before. So let's look here. If we take a data set, this is actually that USA arrest data set that I looked at earlier with the murder, whatever, crime and so forth data set. This is what happens if you PCA it. And what I think is actually kind of interesting is to see what happens if you do an LDA, and this is an LDA with different hyperparameters. But if you, as I told you, if you use that one hyperparameter alpha, you can kind of ramp it. You can see you get a very, very sparse distribution of topics, for any one sample, and you get a kind of cleaner picture. And so that's one of the positives about LDA, why people like that, that technique.